my talk will involve next-generation sequencing for diagnosis and surveillance of emerging infections. I just want to start with some disclosures. I do get research support for pathogen discovery from Abbott Diagnostics. I am also on the scientific advisory board for 2 genomic companies. So for the summary of my talk, we'll start out by talking about what I mean by unbiased metagenomic next-generation sequencing, or NGS, for infectious disease diagnosis. We'll then talk about a bioinformatics analysis pipeline which has been developed for pathogen detection from sequencing data. We'll then talk about rapid pathogen identification using a new technology called real-time nanopore sequencing. We'll go over a few clinical case reports. We'll talk a little bit about applications for pathogen discovery. And then finally, we'll end by discussing a project called the CIAPM project, which stands for California Initiative to Advance Precision Medicine, a way to implement this clinically validated NGS test for diagnosis of infectious disease in hospitalized patients. And the motivation for this is that there is a great clinical unmet need in that for many acute and life-threatening infectious diseases, as you can see here on the slide, including pneumonia, diarrheal disease, meningitis, encephalitis, and sepsis, we really lack a sufficiently sensitive or specific diagnostic tests, or the tests that we do have tend to be delayed. And as a result, we miss up to perhaps 25% of pneumonias, about to 50% of cases of diarrheal disease, and for a disease like encephalitis, up to 80% of the time the cause can remain unknown despite extensive testing in the hospital. It's also been well described that even for well-characterized syndromes such as hemorrhagic fever and sepsis, for instance, up to 20% of the time the cause remains unknown despite testing. So our approach is, instead of using kind of the traditional infectious disease paradigm of one test per target, so for instance, one influenza test to detect influenza, our approach is to use a single nucleic acid sequencing test to detect nearly all microbes. And in principle, because all of these microbes that you see here, bacteria, viruses, fungi, and parasites that cause human disease, are made of DNA, there's no particular reason why in a single nucleic acid test or a single DNA-based sequencing test we can diagnose or potentially diagnose all of these agents. Of course, we're missing one agent, which are prions, which are protein-based instead of being DNA-based. However, we can really detect the vast majority of agents that cause human disease. So our approach is known as metagenomic next-generation sequencing. And it's called metagenomic because the general idea is we want to detect sequences that represent all kinds of genomes, bacterial, viral, parasitic, or fungal, all of these pathogens, microorganisms that cause infectious diseases. However, as you might expect from human clinical samples, most of what you'll be sequencing in a metagenomic approach are probably going to be from the host. And this poses a big problem, which we'll go into in later slides. So the approach is a brute force approach involving random or shotgun sequencing of all of the DNA and RNA, all of the nucleic acid in clinical samples. This enables us to not only detect, but to potentially to actually reconstruct the entire genomic sequence of nearly any and all pathogens at once. What's made this technology feasible over the past couple of years has been the vast increases in sequencing capacity, namely on an instrument like the MiSeq, we can generate about 30 to 40 million reads per run. On an instrument like the HiSeq, we can generate more than 1 billion reads per run. And even on point-of-care smaller instruments, which we'll go into in future slides, such as the Oxford Nanopore MinION, we can still generate about 100,000 sequence reads per run. And the ultimate goal of this is that we would be able to accelerate diagnosis by using these next-generation sequencing tests. And the goal would be to impact clinical decision making. This is a process which is known as precision medicine. And the general idea is that instead of empirically treating patients with infectious diseases, meaning that we treat them without really knowing what the diagnosis is, trying to make a best guess, we are able to implement a comprehensive yet specific test to make a very accurate diagnosis. And by doing so, we could then provide both early as well as targeted treatment that would be cost-effective and actionable. And ultimately, this would result in lower healthcare costs and improved patient outcomes. So the general approach for identifying pathogens on the basis of their specific DNA sequence is shown here in this slide, where we start out with raw sequencing data. We then clean up those reads in a process called pre-processing. We then identify the background, which, as you might expect from human clinical samples, are human reads. We then computationally subtract, after computationally subtracting those background reads, we then identify pathogen-specific reads, reads that may correspond to viruses, bacteria, fungi, and parasites that may be potential signals that these microorganisms are present in the clinical sample. And then this would ultimately result in detection and diagnosis. Now, traditionally, this has been a big computational bottleneck. In particular, two of the procedures, identifying human background reads as well as identifying pathogen-specific reads by aligning these millions of sequences to extremely large databases, has been a big computational challenge that's taken days to weeks. And as you might expect, unlike other fields such as cancer, where perhaps having a delay of a few days is acceptable, in infectious diseases with critically ill patients, time is really of the essence for making a diagnosis. So we would really want a computational pipeline that can actually identify agents within minutes to hours as opposed to days to weeks. So partly to address this challenge, we've developed the bioinformatics pipeline that we call SURPI, Sequence-Based Ultra-Rapid Pathogen Identification. The goal of this pipeline is to analyze millions of next-generation sequences in minutes to hours and be able to provide data that would be interpretable by clinical personnel such as doctors and laboratory directors. And in a way, this directly addresses the computational analysis bottleneck. We streamline the algorithms. We actually use extremely fast algorithms that are orders of magnitude faster than traditional algorithms for analyzing the data. I should also mention that this is not the only pipeline that's available. Other groups have also been working on and have developed pipelines as well. But this pipeline was designed from the get-go to be something that would be streamlined and would enable analysis, within minutes to hours. We also have a clinical version called SURPI Plus, which is in active development. This goes over kind of the process of how we actually do the sequencing and then do the analysis to make an identification. So if you focus on the left side part of the slide, we basically start out with a clinical sample, extract the DNA and RNA, we generate the library, and then we sequence it. And then if going to the right side for the analysis, those are the steps of the analysis. As you can see, we trim the data, we assemble the data into larger sequences or longer sequences called contigs. We then remove human sequences because that represents the background in this particular test. And then we identify pathogens, such as you can see in kind of the third panel on the right, fourth panel on the right, you can see an example of a virus, a bacterium, a parasite, and a fungus. And we do that by aligning the sequences to very large reference databases. We finally are able to classify them, and this becomes very important because you not only want to identify, say, a virus, but you want to make sure that your identification is specific. So we use analytical methods to classify the hits properly, and that way we can kind of report the results in kind of an easy-to-use visualization, kind of web-based visualization tools that we've developed. So I want to focus now on specifically what we do for clinical interpretation results reporting, because this is really a critical aspect of the analysis, in that we not only want to analyze millions of sequence reads, we want to be able to provide data that would be usable by those working in medicine or in public health. So what we do is we do things such as filter sequences that happen to be misannotated in the databases. We are able to tag some of what we call the metadata, the associated data with the sequences, so we can identify, for instance, which of these organisms are colonizers or which ones are more likely to be pathogens. We're able to provide a front-end visualization tool and a web-based interface. We're able to do, as I mentioned before, accurate classification, so we make sure that all of our hits are accurate and precise. And we also have implemented measures to improve processing speed, as well as provide different implementations of the software, such as on a laptop and potentially on the cloud. So I want to go a little bit into the software. So the visualization aspect of the software that we developed is called SURPI Viz. Viz stands for visualization. This is kind of a web-based visualization tool that enables you to look at next-generation sequencing data. One of the advantages of using SURPI Viz is that you don't need to be a programmer or bioinformaticist to be able to run this software. We actually now have clinically certified technicians who are actually running and using this software on a daily basis. this gives you some example of some of the what the data may look like. So what you actually see here, this is a heat map of hits where typically hits are in kind of basically more reddish orange colors, whereas no hits or negative results are shown in green and in yellow. As you can see here, these are basically 8 samples, and the samples are arranged. They're designated 67 to 74, and they're arranged in columns, and what you see on the left side are actually the hits to different viruses. And as you can see, simply at a glance, we can see that sample number 71, for instance, has a strong hit, 108 reads that align to Zaire Ebola virus. And indeed, this was actually a sample from a patient who was infected with Ebola, hemorrhagic fever, or Ebola virus disease. Now, we don't have biosafety for lab. We're not actually, we are processing the sample, but the samples are inactivated before they're sent from Africa to the United States for analysis. But you can see here, this is an example of how, at a glance, you can see, that this is a patient that is infected with Zaire Ebola virus. I want to talk a little bit about CLIA. And this is important because all clinical diagnostic tests need to be done within a certain regulatory framework called CLIA, at least in the United States. CLIA stands for Clinical Laboratory Improvement Amendments. This is a federal regulatory framework for clinical laboratory testing that took effect in 1992. CLIA laboratories are accredited or certified by inspection agencies. So for instance, our CLIA laboratory, which is the UC San Francisco Microbiology Laboratory, is certified by the College of American Pathology. What CLIA does is it sets minimum standards under which all clinical laboratories operate. And it really requires validation and quality assurance for all laboratory tests that are used in clinical care. Now this would include the validation of the sequencing metagenomic NGS assay that I've been talking about, and it's really essential that this be done in a CLIA environment so that we are able to actually implement this clinically and be able to report results on diagnostic results for patients. So what we've done is, in addition to the actual pipeline and the actual protocols, we've managed to move the entire workflow into a CLIA-certified laboratory. And the goal is that everything would be done in a CLIA environment. It would be run by clinically licensed scientists, as well as done in a regulated fashion. The analysis is done as well. So the analysis is not independent of this, but it is rather a critical part of this. And the goal would be to have a test that has a total turnaround time of between 12 hours and 2 days. This shows you kind of the broad applicability of this approach. So what you actually see here, results from analysis of samples from 6 different patients. So 2 patients who are in the first row actually have nasal infection. These are 2 children from UCSF with nasal disease, with actually upper respiratory illness. On the lower part of the slide, you can actually see these are samples from 4 patients with acute febrile illness from Africa. And as you can see on the top 2, you can actually see that we can detect 2 viruses that are linked to respiratory disease in children, Metagenomic and rhinovirus type 78. In addition, from the plasma or blood samples from the patients from Africa with fever and in some cases sepsis, we can identify infection by adenovirus, by a bacterium called Haemophilus influenza, by another bacterium called Salmonella typhi, which is the cause of typhoid fever. This was a patient with typhoid fever, as well as even a parasite. And what you see here is the lower right is a box showing analysis or sequencing analysis of a patient who has acute malaria and is infected with the cause of that, which is Plasmodium falciparum. What I show there are actually just 4 of the many chromosomes that are in part of the Plasmodium organism. So to summarize, ultimately what we want to do here is to provide more timely diagnosis that will result in improved clinical outcomes. And we're aiming really here for a 12 to 24-hour turnaround time. Now, we don't envision that this is really going to be a frontline test that's done in the emergency department, but this would be potentially a second-line test. After you've already done the kind of point-of-care test or the immediate test and the patient still lacks a diagnosis, you could then send this assay, which potentially can be done in a time frame that would be better than kind of existing methods which involve kind of turnaround times that may be within several days to weeks, such as send out molecular testing, even antibiotic resistance testing and culture. I want to give you a few examples here. So this is actually Josh Osborne, and the photo is given to me courtesy of the Osborne family. He was a 14-year-old gentleman who had 3 hospitalizations over 4 months, spent 44 days in the ICU, intensive care unit, had over 100 inconclusive diagnostic tests for different pathogens, had 3 empiric treatments to no effect, eventually underwent a brain biopsy and induced coma for his disease, and at a cost, as you can see there, a significant cost. So we were able to, when he was critically ill, we were able to look at a cerebrospinal fluid sample from this boy. And to make a long story short, we were able to identify within 48 hours that his cerebrospinal fluid, by the NGS assay, was in fact harbored an organism called Leptospira centrosai, which is the cause of a disease called leptospirosis, which is a bacterial disease of the central nervous system. He was cured within 2 weeks after his diagnosis from NGS diagnosis with appropriate treatment, which in this case, ironically, is simply IV penicillin, intravenous penicillin. This is a report that was published in the New England Journal of Medicine. I want to show you what it actually looks like on our new software, SURPI Viz. So you can see here, this was on the left, you can actually see the left column actually is a DNA sample from cerebrospinal fluid from Josh. You can see here that the top hit is indeed Leptospira centrosai, which was the organism that caused his encephalitis meningitis. This is another way to look at the data. This is called a chronoplot, which basically displays kind of the various species and genera and families of organisms that were detected. As you can see here, the majority, in fact, the vast majority of the reads align to Leptospira. This is looking at kind of the, again, the DNA portion of his sequencing data. I want to go over another example because as an example to show that we can not only detect bacteria, this is actually a case of a viral infection. This gentleman was a 55-year-old male with deafness and behavioral change. He had a bone marrow transplant in May of 2013. Mid-October, he developed rapidly progressive hearing loss. He actually went from completely normal hearing to completely deaf within 2 to 3 weeks. Testing for common agents, viruses that may cause this were negative. He was treated empirically with antiviral medications, antibiotics, intravenous immunoglobulins, steroids to no effect. Over the next few weeks, his symptoms became worse. He developed nausea, fatigue, persistent hearing loss, and then depressed, irritable mood, actually a personality change, which was very unusual for the patient according to his wife, and was finally sedated under the UK Mental Health Act. He had an MRI scan of his brain late December, which showed that he had developed some abnormal signal in his midbrain and his thalamus bilaterally on both sides. So he had a brain biopsy that was performed to make a diagnosis. What you see here is actually a slice across his MRI scan, and shown in the white arrows are some hyperintensities that are seen on imaging which are abnormal, which are found in the midbrain and thalamus. So it's clearly he had some brain process, neurological process, a pathologic process that was likely the cause of his symptoms. And so we were able to look at, in this case, brain biopsy tissue from him, and we were able to identify using the NGS assay that this was a case of Astrovirus Encephalitis. When astroviruses are actually emerging viruses, emerging causes of encephalitis. Over the past year now, there have actually been several case reports of astroviruses causing illness, primarily in both children and adults who are immunocompromised. And this gentleman was immunocompromised. And you can see here, what you see on the upper right is you can actually see staining specifically for this astrovirus. He had a strain that was called the VA1 strain, or the HMOC strain. And you can see here that it's shown there in pink is kind of a phylogenetic analysis of the sequence from his virus. And you can see here it fits in basically with a particular clade that has been linked to encephalitis, encephalitic disease. Now he was an adult primarily the other cases of this astrovirus have been found in children, because actually astroviruses are actually common viral causes of viral diarrhea in children. It's only recently that now these astroviruses have been linked to potentially fatal encephalitis or brain infections in adults and children. What's interesting is that the two closest relatives, animal relatives to this virus or this particular clade, are mink astroviruses, shown there as well as bovine astroviruses, which are, and you can see there that they're actually closely related by phylogenetic analysis. What's interesting is that in both minks and in cows, that these viruses have been shown to actually cause encephalitis in animals as well. So he was started on antiviral therapy and intravenous immunoglobulin, but unfortunately there's no treatment for astroviruses. So despite treatment, he passed away 4 months after his diagnosis. However, this is a point that I would like to make, is that had there been a treatment, the fact that we were able to use a comprehensive test that was able to pick this up might have made a difference had there been an effective treatment for this disease. This is another case. So we went over a case of a bacterial infection, a viral infection. I want to now talk about a parasitic infection. This was a case of hemorrhagic encephalitis, which actually is as bad as it really sounds. I mean, it really is bleeding in the brain as a result of an infection. This is a 15-year-old girl with type 1 diabetes who had 7 days of headache, vomiting, arm weakness, and confusion. She developed these very large hemorrhagic lesions on MRI scan. You can see here shown as one slice where you see the entire area in white is basically a lesion. She had extensive diagnostic testing that was negative. And the clinicians who evaluated her when she came in thought that this was a classic presentation for what we call the hemorrhagic form of an autoimmune disease called acute demyelinating encephalomyelitis. So she was actually given steroids, which is appropriate if she had an autoimmune disease. Unfortunately, she did not, and she actually had an infectious disease. So after 5 days, she actually got worse and was actually markedly worse, so had a brain biopsy that was done. And it turns out that this was a case of Balamuthia encephalitis. So for those of you who are probably not familiar with this bug, this is one of the causes of what is known colloquially as brain-eating amoebae. This is a disease which is actually acquired by exposure to the soil. She may have acquired this while playing in the playground, for instance. So she actually had, by the sequencing test, more than 6,000 reads that aligned to a specific sequence from this parasite, Balamuthia mandrillaris. She also had the brain biopsy analysis, pathologic analysis, which showed that numerous examples of the amoebae, you can see here kind of in a blow-up. This is an example of amoebic cell. The cell is actually in cyst form in the brain tissue. If you actually look on the right, that's actually the cell kind of when in the vegetative form when it's actually active. And unfortunately, the diagnosis was made by day 7, but despite combination antibiotic therapy, antifungal therapy, anti-amoebic therapy, she developed intracranial hypertension, cardiac arrest, and died. So we actually wanted to go back and see, could we have made a difference? If perhaps could we have diagnosed it earlier, and if so, would it have made a difference if we could have made the diagnosis perhaps on day 1 when she initially came to the hospital? So we actually took a look at her cerebrospinal fluid taken at day 1. And what was quite interesting there was the answer is actually no, based on what was available at the time with respect to the reference databases. If you recall, the way we're able to make an identification here is we generate millions of reads, but then we align them to a reference. Now, so therefore, your test is really only as good as your reference database. And it actually turns out that there are very, very few sequences to Balamuthia that were existing in the reference database as of 6 months ago. Now, in the meantime, my laboratory had actually been working on the genome sequence, the entire genome of Balamuthia, which is about 50 megabases in size. And what's interesting is once you include that particular genome in the reference database, then you actually are able to detect the bug, detect sequences from this particular organism on day 1. As you can see on the left side where we detected 20 reads from the cerebrospinal fluid had there been the genome in the reference database, but 0 given with the existing databases. And this really highlights that microbial reference sequences are really critical for this procedure. I want to switch gears a little bit. I want to move into kind of a new technology that we've been working with for diagnosing emerging infections. And this is called the Nanopore, and the technology is called Nanopore sequencing. What we've done here is we've actually taken the same protocol that we've been using on an Illumina instrument. That's the traditional protocol for diagnosing this disease, and we've been moving it on to the MinION and the Oxford Nanopore. The advantages of putting this on a Nanopore is, unlike the MiSeq, which has a 24 to 48 hour turnaround time, we're actually able to go from sample to getting an answer within under 6 hours. And what the Nanopore really is it's kind of a USB size, slightly larger than a USB size stick that enables you to do effectively real-time sequencing analysis. So the difference between that and the more kind of commonly used technology, which is Illumina sequencing, is that you can actually analyze the data as soon as you start sequencing it. Whereas for Illumina sequencing, you actually have to wait until the data is fully sequenced before you can analyze it. And that's why there's such a big difference in the time to initial detection, under 6 hours for the MinION, Oxford Nanopore, and more than 20 hours at a minimum for Illumina sequencing. So this gives you an example where we are able to detect viral reads in this case, from a plasma sample from 2 patients who have acute fever, acute febrile illness. We were able to detect within 6 minutes that one patient was infected with Chikungunya virus, and on the right you can see within 8 minutes we're able to detect infection by Ebola virus. And again, I want to stress that we're using metagenomic NGS. In other words, we're not targeting specifically Chikungunya virus or Ebola virus. We're simply sequencing everything in the sample, doing real-time sequencing analysis, and seeing if we can detect the organism as soon as possible. What also was quite impressive by some of our work is that basically that we're able to get kind of near complete genome coverage with really high confidence sequence assembly. You can see here that this is showing you Chikungunya virus, where we're able to basically, with very high identity, be able to identify Chikungunya virus, and if you have sufficient coverage, you can actually identify and sequence the organism accurately. I want to talk a little bit about Chikungunya virus, because Chikungunya virus is an emerging viral infection. This is a study that we just published, actually, in Emerging Infectious Diseases in 2015. These are cases, case epidemic curve of Chikungunya virus in Puerto Rico. This is done in collaboration with the American Red Cross. And as you can see here, in 2014, there was actually a very large outbreak of Chikungunya virus. It's actually, over the past several years, has come into the Caribbean. It's basically an imported disease from Africa and areas of Asia. Now it's actually in the Western Hemisphere for probably the second time, but now it's more recently emerged in the Western Hemisphere. And what we did is we decided to use sequencing to identify, to look at 3 cases shown there in the red asterisks. So what we did is we actually did both PCR as well as next-generation sequencing analysis of Chikungunya virus positive blood donors. And what's interesting is that we can not only detect Chikungunya virus by a PCR, polymerase chain reaction, but that given the high titers of the virus in infected patients, we can actually assemble the genome, the entire genome of this virus. And why would you want to do so? Well, there are a couple of reasons. One reason for identifying the genome, as you can see here in the next slide, is that getting a sequence of the genome enables you to do phylogenetic analysis, to place exactly where does this virus fit in the NGS data. And you can see here that really it fits in a clade, as you might expect. It fits in a clade that's been linked to all consisting of viruses from the Caribbean. Chikungunya viruses in the Caribbean. So you can see here that the Puerto Rico strains that we sequenced here, using either the MiSeq or the MinION Nanopore, are related to previously sequenced strains in the island of Saint Martin, Martinique, as well as the British Virgin Islands, all of which are part of the Caribbean islands. In addition, although I don't show it here, we can also do a technique called molecular clock analysis. We can actually use this phylogenetic data to be able to predict when did this virus emerge. And by doing so, we were able to actually show that this virus emerged in the Caribbean in probably in early 2013. So we're able to kind of date the emergence of this virus in the Caribbean to 2013 and in Puerto Rico to early 2014. You can refer to the publication for more details. So this is basically gives you a kind of a look at the MinION. You can see here that the MinION is a little bit larger than a USB stick, but the general idea is that the MinION consists of a flow cell that has consists of a flow cell with pores, and what happens is as you inject the library, as the DNA flows through the pores, you can do real-time sequencing analysis on your laptop. Okay. I also want to show that this is not only the case, the phylogenetic analysis is not only the case for Chikungunya virus, but also works for Ebola virus. And as you can see here, this is basically using either the MinION or the MiSeq instrument, we're able to identify this particular Ebola virus strain as a strain that originated from Central Africa, from the Democratic Republic of the Congo, as you can see here in pink. So this is not the strain which is, in fact, did not come from the same outbreak as probably the more widely known West African outbreak, which is shown here in yellow, which was the outbreak in the countries of Guinea, Sierra Leone, and Liberia. So we can, by phylogenetic analysis, by sequencing, we can not only detect the agent, but do the phylogenetic analysis to be able to precisely identify the strain of the pathogen. I want to go a little bit more into Ebola virus. So we are currently using this technique of sequencing to look at Ebola virus samples collected from Central Africa. And it's our goal that we can use the metagenomic NGS test as a way to improve Ebola virus diagnostics. So what you actually see here, there was an outbreak of Ebola virus disease in Congo in 2014, and actually happened in 2 provinces in the 2 villages in the Democratic Republic of the Congo. What you see here are actually 8 samples from this first 8 cases in this outbreak, including the index case. And as you can see here shown in the red, that metagenomic NGS was as sensitive as both conventional and real-time PCR, which is the gold standard testing for this disease as in the diagnosis of Ebola virus disease. We're also able to develop some enrichment methods where we can basically enrich samples so that we can be able to sequence the entire genome of the virus directly from patient samples. I also want to talk about some other aspects of the analysis. So the NGS analysis, the NGS test, the next generation sequencing test, as I said before, is a great way to identify pathogens in kind of a comprehensive and unbiased fashion. But what's also interesting is that you can actually do what we call host response analysis. You can actually look at the human portion, which we normally throw away the human part. But our idea is that we can actually use the sequencing data that corresponds to the human response to be able to identify the disease. So we're currently doing a study where we're actually looking at samples from patients with different types of hemorrhagic fevers, different types of febrile illnesses from Africa, so either Ebola virus, another virus called Lassa fever virus, as well as malaria, to see if can we identify potentially markers that by sequencing, can we identify human markers or markers of gene expression that can help diagnose potentially the disease. Now this is of great interest. Part of the reason this is of great interest is that even the most sensitive PCR tests can't really detect Ebola virus in some cases of early disease or before a patient has symptoms. And it's our hope that potentially by doing this technique, we can actually identify and diagnose patients with Ebola even before they have symptoms, which would, as you might expect, would probably be very helpful with respect to outbreak containment and infection control. So we were able to do this study and identify that there were hundreds of unique differentially expressed genes called DEGs that were identified in Ebola virus patients. We also identified reiterated many of the existing pathways that have been linked to Ebola virus infection. And we're currently using this information to actually develop biomarker panels. So the idea is using the sequencing to then develop biomarker panels that will help you diagnose or differentially diagnose Ebola hemorrhagic fever and distinguish it from other causes of hemorrhagic fever in Africa. We're currently working with 2 groups in West Africa to be able to implement this potentially as a sequencing test that could be done kind of in the developing world, in point of care laboratories in the developing world. This gives you another example where the same approach doesn't necessarily apply just to Ebola. This is a manuscript that we recently submitted where we're able to do a signature identification for acute Lyme disease. Lyme disease is also another infectious disease where diagnostics is a big problem. Currently, the gold standard test is two-tiered serological antibody testing, which is really only good kind of 2 to 3 weeks after the illness and doesn't really, after you develop kind of symptoms of Lyme disease, the fever and the rash, and are really not useful in the acute setting. So we're hoping on using this to develop new tests that can diagnose the disease in the acute setting. And we're able to show here that you can see that the classifier panel, using the gene analysis, using sequencing, is specific to Lyme, as you can see here in kind of the blue dots. This gives you another example of where we took the samples from the hemorrhagic fever study. We identified then a 25-gene biomarker panel. We narrowed it down to 25 genes, and we then are able to show here that those 25 genes appear to be fairly specific for diagnosis of Ebola virus. What's interesting here also is that you can see of the 5 samples that we ran this panel through, one of them is highlighted in the asterisk. That was actually a patient that was PCR negative. So that was actually a patient that was, before the virus was actually detectable in the patient's blood, we were able to identify that that patient at least did have a signature of acute hemorrhagic Ebola virus infection. I want to talk about another emerging virus because part of the theme of this talk is discussion of applying sequencing for emerging viral detection. I want to talk here about Enterovirus D68. This is a very interesting virus. It's a respiratory virus that was initially discovered in the 1960s. We recently published a paper this year, actually, where I used sequencing, the same sequencing testing, to be able to analyze samples from patients that were infected with Enterovirus D68. The other thing that has been linked to Enterovirus D68 is an acute flaccid myelitis syndrome. So acute flaccid myelitis is a disease where mostly children, shortly after getting an acute febrile illness, they then develop kind of a flaccid paralysis. And this can happen in one limb, it can happen in both limbs, in both arms and legs, and eventually can even progress to involving respiratory failure. And this was quite alarming because it happened in previously healthy children, a small subset of children that had developed an acute febrile illness. The other thing that was also interesting, that the timing of the cases of this acute flaccid paralysis appeared to coincide with that of a very large respiratory Enterovirus D68 outbreak that occurred last year throughout the United States in 2014. The months were mainly between August and December. The question has always been, is Enterovirus D68 the cause, or potentially the cause, of acute flaccid myelitis or acute flaccid paralysis, or was this simply a coincidence? And this was a difficult question to address, both by hospitals and public health agencies, because really we don't really have routine surveillance for enterovirus infections. So this was a big mystery because it was a nationwide outbreak of a respiratory viral infection, Enterovirus D68, that sent children to emergency departments. And yet we don't know why some children that had a respiratory infection, had Enterovirus D68, eventually did develop kind of acute flaccid paralysis. And what we want to do is we want to use the sequencing tests to see if there really was an association. So some of the key questions we wanted to answer here, and I should preface this by saying that we didn't answer all of them. In fact, there's ongoing research to try to address some of these. But one is that, is Enterovirus D68 infection really associated with acute flaccid myelitis? Was there something special about this virus that was detected in about 50% of children with acute flaccid myelitis that had Enterovirus D68 infection? So the other thing that was interesting was that only 50% of the children that had Enterovirus D68 infection had 50% of the children with acute flaccid myelitis had Enterovirus D68 infection. And so the question was, what about the other 50%, who didn't have Enterovirus D68 and yet still had acute flaccid myelitis? It made the link much less convincing. The other question is, we wanted to identify, could we use this metagenomic NGS test to perhaps identify an alternative etiology? As I already discussed, one of the key advantages of this test is that we don't bias our detection. So potentially, could we have detected perhaps another virus that may have been missed? We also wanted to determine is whether or not we could detect the virus in cerebrospinal fluid or blood, because prior to this point, it had only been, and in fact to date, it primarily has been detected only in respiratory secretions and not in cerebrospinal fluid or blood. And then finally, we wanted to address some of the questions of why is detection even in respiratory secretions unreliable. So as opposed to kind of going over the case in detail, I just want to highlight a few things. This was a case where we looked at cases in 2 states, California and Colorado, of which the vast majority of these cases have been described. We looked at a total of 37 patients from California, 16 from Colorado. As you can see, not all of them had Enterovirus D68. In fact, only the ones that are shown in red actually had, were infected with Enterovirus D68. And as you can see here, the ones with Enterovirus D68 coincided very well with the outbreak. But of course, given that we don't see Enterovirus D68 in all of the cases, this still raises sort of like a question of, is this really associated with acute phacoamyelitis? So one of the key findings that we actually made in this study, which was published in Lancet Infectious Diseases, is that we actually discovered that all of the cases of Enterovirus D68 in children who had acute phacoamyelitis came from a specific clade. And this is a clade which is a group of viruses, as you can see here in the phylogenetic tree, which we call clade B1. And what you see here in red are actually all of the cases that we sequenced of acute Enterovirus D68 from acute phacoamyelitis patients. What you actually see in blue are actually the cases that are from pediatric patients that only have respiratory disease. And it really highlights the fact is that this virus it's not as if this clade was only found in viruses in children that had acute phacoamyelitis. It was really found, and it has been found quite commonly, perhaps mostly in children that have respiratory disease only. So only a certain subset of these children actually, and a very rare subset, actually developed acute phacoamyelitis of children infected with Enterovirus D68. So these are some of the key questions. I don't want to kind of go over the entire study, but I just want to just answer some of the questions were answered and some were not. So one is that we did find that there was an association with acute phacoamyelitis. We found it in more than 50% of patients that had AFM. It was really the only agent that we found actually not the only agent, but the majority agent that we found in nasal secretions. We actually did find it in blood, actually, from one boy that had it in his blood, in his respiratory secretions, and also in his stool. Interestingly enough, we did not see it at all in cerebrospinal fluid. And we hypothesized that perhaps this is because many of the samples that were collected or are collected late in the course of illness, where a child will develop acute phacoamyelitis, but that could be easily 1 to 2 weeks after the acute infection, the acute febrile illness. So we suspect that many of these samples were probably collected too late. Although it's also quite interesting, by using our metagenomic NGS assay, we didn't detect any evidence of any alternative etiology. So with respect to infectious etiology, so far, Enterovirus D68 is still our best candidate. I just want to go over some recent unpublished data. This is done in collaboration in the laboratory of Dr. Ken Tyler at the University of Colorado. What we did here is we wanted to demonstrate a mouse model of Enterovirus D68 infection. So what we did is we actually took 2 samples from the clade B1 strain that was found in the children with acute phacoamyelitis, shown there in U.S. Missouri, U.S. Illinois. We also injected with the prototype strains of the virus. And to make a long story short, we've been able to reproduce this in a subset of mice that are infected, inoculated with this virus. We can actually we can see a limb paralysis. In fact, symptoms that resemble that seen in the children. And although this is very early data, it is actually quite suggestive, and confirmatory studies are underway. So I want to summarize the points that I've made. One is that I believe that metagenomic NGS, or next-generation sequencing, is a very promising approach for diagnosis of infectious disease. New clinical bioinformatic pipelines such as SURPI can greatly improve computational turnaround times and make these technologies accessible for clinical and public health laboratories. NGS for pathogen detection discovery is only the first step. As you can see with the Enterovirus D68 story, the hard work is yet to follow. And finally, I feel that many regulatory and technical challenges remain for this technology, but they have evolved to where we can really implement them clinically and for use in both clinical medicine and public health. I want to finally end by acknowledging all of the collaborators and members of my lab who are involved in this work. They include the members of my laboratory who are shown in red, collaborators, especially acknowledge University of Colorado for the Enterovirus D68 work, and others, as well as funding by the NIH with the UC Discovery Award, an Abbott Pathogen Discovery Award, and the California Initiative to Advance Precision Medicine Award. Thank you very much, and I would be happy to take questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, what is the approximate cost and next-generation sequencing diagnosis approach per patient?
That's a very difficult, it's a great question. It's very difficult, and it probably takes a nuanced answer for which I don't have a huge amount of time. I can tell you that with respect to the actual laboratory cost, it's really the cost of the sequencing, and it's the cost of having, say, if you want to use this clinically, of having a CLIA scientist or a clinically licensed technician to run the assay. Now, we've actually estimated that, at current kind of market costs, that the actual cost of running the assay can be as low as about $300 to $400. Now, of course, in practical terms, it may be much more expensive given issues of having it run in a clinical laboratory, et cetera. But with respect to the actual cost, is a couple of $100s, meaning that it is more expensive than, say, a PCR or polymerase chain reaction assay done in a laboratory, but actually not too much more expensive. I would say probably 3 to 5 times more expensive than a PCR test, which, as you know, only looks for one or a few agents.
Okay. So next we have, please kindly differentiate between emerging and re-emerging infections, because your topic today was centered on emerging infection, while you are also discussing re-emerging infection.
This is, again, a great question. Really for, so it depends on what you define as emerging infections, for instance. You may argue, for instance, that virtually all of the infections or kind of the outbreaks that have occurred over the past, or the vast majority of them that occurred over the past 10 years are really re-emerging, in the sense that, so to give an example, many would consider Ebola virus to be an emerging infection, and yet, Ebola virus was discovered more than 30 years ago. And this is really a re-emerging pathogen. Now, with respect to emerging infections, those are usually caused by truly novel pathogens, or novel agents that have not been seen before, at least in humans or in the human population. So some examples of that would include, for instance, SARS coronavirus and MERS coronavirus, potentially avian influenza H5N1, et cetera. But arguably, most of the infections that we're concerned about that may cause outbreaks are probably re-emerging, in that we already know about them. It's just that we don't really have really broad diagnostics that can kind of detect all possible agents at once. Although one thing I do want to highlight is that this particular approach, the use of NGS, next generation sequencing, is broad enough so that we can detect, for instance, sequences from very divergent pathogens, usually viruses, that may differ significantly from things that are present in reference databases. So a good example of that is where we use this to identify a new hemorrhagic fever virus, or a putative hemorrhagic fever virus called Bakongo virus, BAS-CONGO. And I would recommend you take a look at it. We published that in PLOS Pathogens in 2012, where we used the technique. And that would arguably be called an emerging infection, because that virus had never before been identified in either humans or animals.
I would like to thank you, Dr. Chiu, for your presentation. Do you have any final comments to share with us?
Sure. I just want to end by saying that there's a lot of interest in this technology. I do think that there are many challenges that remain, and really primarily there are really huge bioinformatics challenges, in the sense that, as I showed you with the Balamuthia case, that really this test is only good as the reference databases. And I am encouraged, though, by the fact that there are now efforts to really expand and really make accurate the databases that will be available for doing this type of sequencing analysis. So I think that the generation of these databases will go in parallel with development of the technology itself. The other barrier I see is that of clinical validation. You need to show that this technique can be used clinically, and by using clinically, you have to show that it's accurate, it's sensitive, it's specific, that you get results that can be interpretable by physicians. And because of the computational challenges and the bioinformatics challenges and the technical challenges, this has been difficult. But I think that over the next 10 years, what you're going to see is that all of the hard work with development has already been done, and now it's really going to be time to implement this practically.
Well, thank you, Dr. Chiu. Today's webcast will be available for on-demand viewing through February 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.